Welcome to our video on conventional gene cloning. We will explore how scientists clone the factor VIII gene, which is crucial in producing a protein known as coagulation factor VIII. This protein is essential for blood clot formation, which helps stop bleeding by sealing damaged blood vessels after an injury, thereby preventing excessive blood loss. So let's get started. Firstly, identify and isolate the factor VIII gene from the cells by removing the cell debris through centrifugation and purification by phenylchloroform extraction. Secondly, select a plasmid or another vector to carry the factor VIII gene. The vector is cut open using restriction enzymes that create compatible ends for gene insertion. Thirdly, insert the isolated factor VIII gene into the prepared vector using a process called ligation, where the gene and vector are joined together by the enzyme DNA ligase. Next, introduce the recombinant vector containing the factor VIII gene into bacterial cells, typically Escherichia coli, by a process called transformation. This allows the bacterial cells to take up the vector. After that, grow the transformed bacterial cells on selective media that contains antibiotics. Only the bacteria that have taken up the vector with the antibiotic-resistant gene will survive. We can screen these colonies to identify those that also carry the factor VIII gene. Next, grow the selected bacteria in large quantities. As the bacteria replicate, they also replicate the recombinant vector, thus amplifying the factor VIII gene. Lastly, extract and purify the recombinant plasmid containing the factor VIII gene from the bacterial cells for further use, such as in gene therapy or research. These steps ensure that the factor VIII gene is successfully cloned and can be used for various applications, including the treatment of hemophilia A.